I'm Jason Roberts. The purpose of this section is to just talk about, and if people aren't interested in coming back in, that's okay. My section is just to talk about the different types of therapies that are going to be discussed. So the things that I'm going to be talking about, so RNA knockdown, we're not discussing that today. In the sessions, but it is a therapy that will be emerging in time. And then the other two topics that we're going to look at, so gene transfer, that is the main gene-based technology that's going to be discussed in the sessions. And then there's also a session later this afternoon on gene editing when Intellia Therapeutics will present. All right, so RNA knockdown. There's two different types to knock down mRNA. So the two different modalities are antisense oligonucleotides and small interfering RNA, or siRNA. In terms of how these work, so these are prime time now in clinical medicine. There are drugs that people use now with these modalities. So siRNA and ASO, the way that they work is essentially they're ways to just knock down the mRNA before the mRNA is able to be translated by the ribosome. So ultimately, the protein is not expressed. So I won't go through the kind of nuanced details that's different between siRNA and ASO, except to say that these molecules bind with the mRNA. And I have to say, it's unfortunate that people aren't. Anyways, okay. So the mRNA isn't ultimately able to be expressed. So what diseases are potentially amenable to RNA knockdown? Ideally, there are potentially ideal targets are pathogenic gain of function variants and dominant negative alleles. So this is where. And if you're using this approach, you have to be losing a portion of gene expression must also be tolerated. So this isn't going to be good for a lot of inherited arrhythmia and cardiomyopathy conditions where, so for gene transfer, you're treating these conditions because they've lost part of their the gene expression. Here you're so long QT syndrome, the major subtypes, it's not going to work for ARVC, ACM, or PKP2, DSP, LMNA. Those things aren't going to work. But where they may be potentially helpful are things like phospholamban cardiomyopathy, where you seem to be able to lose about 50% of phospholamban and potentially TMEM43 as well. They're particularly ideal when genes not needed at all. So where these have emerged first are for lipids. PCSK9 is a gene that is important in cholesterol metabolism. And it doesn't seem like adults need any PCSK9. And in that context, there's an siRNA that is now being used clinically called inclisiran that is given once every 6 months and knocks down PCSK9, both alleles. It's targeted to bring down both. Another area is in lipids is Lp(a). So this also predisposes to atherosclerosis. It doesn't seem like you need Lp(a) either. So this was an ASO from Ionis Pharmaceuticals that now Novartis owns it, but works really well at reducing Lp(a) expression, and this is more recent data with siRNA. So is this cardiac prime time yet? Not yet. Delivering these molecules, they're able to get it to the liver very well. They're working on developing it, delivering it to the heart. It depends on how you tag these molecules in terms of where they go. So there's promising preclinical data. So this was a phospholamban mouse model where they developed, so as people mentioned, I think already, so phospholamban, there's an R14del variant that accounts for a large proportion of phospholamban cardiomyopathy. So they developed an ASO specifically for R14del. Knock that down, you have 50% of normal phospholamban expression from the normal allele, and the mice seem to be completely protected after treatment for that. So, and I think the same potential thing could happen with TMEM potentially, but They're still working on getting those to the heart, but it's coming. So gene therapy, so this is the main thing that I wanted to focus on today, because these sessions that we're going to have, most of them are on gene therapy or gene transfer. So the nomenclature it varies a lot, but I view gene therapy as gene transfer. You're delivering a gene to an organ with a viral vector. So this is a general kind of overview of it, and I'll highlight in just a minute. A key thing with gene therapy is the delivery vehicle. Are you able to get the gene effectively into the cell, and is it ultimately safe? So the vectors that are going to be discussed today are all AAV, adeno-associated virus. They're taken up by the cell, then into the nucleus, and then generally established as an episome, so they're not integrated into the genome, and then the wild-type protein or the normal protein is expressed, and if a disease has developed because you're missing one copy of the gene, this is a way to potentially cure the condition. So yeah, I kind of went through this. So delivering a gene with a virus into a cell is called transduction. So transformation is getting DNA material on its own into a bacterial cell. Transfection is DNA material into a eukaryotic cell without a virus. Using a virus, it's called transduction. And it seems to work overall pretty well. There's limitations, so size limitations using AAV. The gene can't be too big. So Titin cardiomyopathy is obviously the major cause of DCM, but Titin's too large to get in an AAV vector. And I went through in terms of what happened. So I mentioned in terms of what diseases are amenable to gene therapy. So you've lost one normal copy of the gene, and you're replacing it. What are not amenable to gene therapy? So gain of function variants and dominant negative as well. So importance of the delivery tool. So we'll hear a lot, I think, from our industry guests here today about their tools. This is important. So gene therapy, back in the 90s, everyone was really excited about it, but it had to be shut down. Back then, they were using adenovirus, which is different than adeno-associated virus, and there was deaths because of dramatic immune responses to these viral therapies. So fortunately, things have come a long way over the past 25 years. The ideal properties of a delivery vehicle, so low immunogenicity and efficient organ-specific delivery. So AAV does not integrate into the genome. There's lentiviruses, which are not going to be discussed today, that are mainly used in the setting of hematology, do integrate into the genome. If they integrate into an appropriate part of the genome or potentially have a very active promoter, there's the potential for induced cancer. But fortunately, AAV does not go into the genome, and that hopefully that we won't see those kind of things coming up. So we can kind of, so AAV, not adenovirus, is what we're going to be hearing about today. And yeah, it's not known to cause human disease, and after it gets into the cell, as I showed in the diagram, it gets established as an episome, not integrated into the genome, and then it expresses the protein. So in terms of considerations for AAV-based gene therapy, and this is just kind of a broad talk, so the companies don't need to go through things. So patients can't have AAV antibodies. So there's a portion of patients that have AAV antibodies. They're not eligible for these therapies, and you can only get AAV therapy once. So after you've had it once, you'll have antibodies, and then would subsequently mount an immune response. So yeah, there's been lots of excitement in this area. So we're going to hear a lot about, I think, PKP2 ARVC today. So this is from some of the companies as well, in addition to some academic groups, talking about how PKP2 gene transfer can potentially cure ARVC. We're going to hear about Danon disease, getting LAMP2 into cells can potentially fix this condition. And this is what's farthest along. So Rocket will be showing human data for their Danon gene therapy program. And this is just in terms of arrhythmia, the preclinical stuff that is not related to the companies, although it's somewhat related to Solid's presentation later. So this was just in the setting of CPVT. 10 years ago, Sylvia Priori was showing us that if you get Cask2 into Cask2 CPVT, it can cure them. I won't, just in the interest of time. So Mike Ackerman, who we all know, has a very, very creative way to try to treat a lot of conditions with a type of gene therapy. One of the challenges with these conditions is that you have many, many different variants potentially for DNA editing. How do you address all the variants? He has this approach called suppression replacement, where you deliver DNA such that there's a small hairpin molecule that will knock down all of the endogenous genes. So if it's long QT1, knock down all of the endogenous KCNQ1, so both the disease allele and the wild type allele that's endogenous. And then the delivery vehicle also contains a normal KCNQ1 that will completely replace the patient's KCNQ1. So that could potentially be used for a lot of different conditions. So that's AAV. So that's the main gene therapies. That's the main thing that we're going to be hearing about in subsequent sessions. And just in terms of brief prep for the Intellia session, so CRISPR. So I think everybody's hearing about that now. So this is here in humans now. It's being done, and the first CRISPR-based therapy was just recently approved for sickle cell in the United States. So how does CRISPR work? There's a few different versions of it. So what Intellia will be talking about is version 1.0. It's a double-stranded DNA break that essentially destroys the gene effectively or eliminates expression. You get a double-stranded DNA break, something called non-homologous end joining. The cell essentially panics, puts it back together, and usually results in a frame shift variant that ends up resulting in nonsense-mediated decay and lack of, and you don't get the protein expressed. So I'm not going to talk about their programs. I'll let them do it, but their programs are essentially knocking down a gene with CRISPR. There's newer types, base editing. It's like a pencil eraser. You can change a C to a T and an A to a G. And then prime editing is the one that has the potential, most potential potentially, but it's the one that's earliest along. It's like a word processor. You can put anything in that you want and take things out as well. And this also, there's a version, an adaptation that involves an integrase that enables you to get a whole gene potentially into the genome in an effective way. And then there's epigenetic editing. I won't go into that. So I think we'll just finish with that. CRISPR, they get the material in with something called a lipid nanoparticle in general, not with an AAV. So anyways, that's just, we felt it was just important to just provide an overview of things that are going to be talked about today, just that each company doesn't feel the need to run through it. So the main area is gene transfer, so generally delivery of a wild type copy of the gene, and helpful for correcting pathogenic haploinsufficiency. So I think I'll leave it at there, and then hand it over to the moderators and speakers for the next session.